Hi, this is Miss Litton, and this is my wonderful Period 1 APBio class. Say hi. Hi. All right. Oh, I can't see it. All right. So last class we started talking about the neuron and how the neuron functions. And we talked about the neuron already having a resting potential. Resting potential is generated by three things. My friends, what is it? Boom, right there. Sodium potassium pump, and what does that do?
It makes like
Three sodiums out for every two potassiums in. And that happens along the length of the neuron, right? Okay, so there are more positive charges outside than inside. Perfect. That's one thing that contributes to the resting potential. And the sodium potassium pump is working all the time. It doesn't wait till the nerve fires and then starts to go. It's always working. Okay? What's the second item that contributes to the resting potential? The what? Ions. Where is everybody else? Okay, I got one student. She's awesome. Maria, go. But there are negatively charged what? Ions and proteins all along the inside. That's number two. What's the third thing? Potassium tends to what? Leak back out. Okay, now this sets up your resting potential. So the outside is positive and the inside is negative when a neuron is resting, and it's ready to fire. So you can harvest that because you can have some sort of neurotransmitter, perhaps, or stimulus. Bathe right here. What's this part of the neuron? The dendrites, which starts to open up sodium gates, which allows some sodium to come in. If enough comes in that it reaches the threshold, then it'll trigger all the gates to open like dominoes that you send off. And it's like, brrr, right? Brrr, there we go. Along the whole length. That is your action potential. That's how you get from point A to point B. Okay? And there at point B, you can release a neurotransmitter which can stimulate the next neuron, muscle, or gland. Now, if I want that neuron to fire again, because the charge has just been reversed along the length of it, now because the sodium came in, the inside is what? Positive, and now the outside is negative. That reversal of charge goes along the length of the neuron. My quick fix is what? Potassium to go back out. That fixes it right away. And so the neuron can fire again because these are voltage-gated channels, and now the outside is positive again and the inside is negative. But if we keep fixing it in that manner, pretty soon all of our sodium is going to be inside and all of our potassium is going to be outside, and it's not going to work. So who's our long-term fix? The sodium potassium pump. I more than likely gave you an analogy about cleaning your room. Did I not? Yes. Okay, good. All right. So when we look, and there are pictures in your book kind of showing it to you in sequence, but if you take a look at this diagram right here. Okay, let's see if we, and this was it in your ed puzzle? Yeah. Okay, good. So when we take a look at this diagram and we see, okay, right here, this would indicate the resting potential. Okay, resting potential, it's negative inside relative to the outside due to the sodium potassium pump, negatively charged ions, and leaky potassium gate. That's what sets it at this resting potential. But now it's becoming less negative. It's still negative, but it's less negative than before. What would cause it? What would induce it to be less negative? Sodium starting to come in. Why would sodium start to come inside the neuron? Because some gates were left open and sodium went from a higher concentration to a lower. What opened up those gates? It could be a neurotransmitter from the last neuron that it synapsed to. It could be, a receptor, some sort, a receptor cell that's triggered. But all of a sudden, sodium's starting to come in. And if enough sodium comes in and it reaches what's right here? Threshold, the light is green, and all of a sudden, then all the sodium gates are opening, and it becomes less and less negative and less negative. In fact, it actually becomes what? Positive. So that, along the length of the neuron, where it becomes positive inside, okay, that's the action potential. That's how a nerve fires. okay? So that's the depolarization of the neuron, okay? Sodium comes in, it depolarizes the neuron. Now, to restore it, the repolarization, to make it negative again, what's going on? What? Say. Louder. Potassium is what? Going out. Okay? Here sodium was going in. Here potassium is going out. Out, and it actually even overcorrects and becomes super negative. Okay? It goes actually, if you look where the resting potential is, it overcorrects, becomes very negative, and this is what helps keep the action potential traveling in one direction. Otherwise, you'd be at a neuron and it could go meh and go both ways. Okay, what would this be akin to in our circulatory system, keeping the blood going in one direction? Valves. You see the big picture? See the connection? Valves keep the blood going in the same direction. This overcorrection right here keeps the action potential going in one direction. So on your notes, go to your notes. Where did I leave off? Because I have no idea. Okay. Okay, action potential is an impulse, and it's starting at the dendrites and ending at the synapse. It's a chemical message, and it begins by opening the sodium gates. Opening the sodium gates. B, sodium gates open, and sodium comes rushing into the neuron. And sodium comes rushing into the neuron due to facilitated diffusion. Due to facilitated diffusion. Sodium comes rushing into the neuron due to facilitated diffusion. Slate, could you please explain what is facilitated diffusion?
I got powder in my brain. That's carbohydrate. Something else.
So did they say something about going from a higher concentration to a lower concentration through a protein channel? Okay. Does it cost any energy? No. Okay. You're just using the gradient. What did cost energy? To set the gradient up, right? That cost energy. Okay. The inside becomes temporarily positive, which triggers the next neighboring sodium gate, and so on and so on and so on. This reversal of charge along the length of the neuron, little letter I, is called the impulse. It gets the message from one end of the neuron to the other end. The repolarization, or the return of the resting potential, in the short term, what happens in the short term to get it back to its resting potential? Perfect, write that down. Potassium gates open and takes sodium's place outside, counteracting the change. Potassium gates open, taking sodium's place outside, counteracting the change. I know, right? All right. And then the long term is the
Sodium potassium.
perfect. Write that down. Sodium potassium pump. That's what I thought I heard over there. All right, my friends. Today, it was suggested. You don't have to do it. Wait, I gotta reset this thing. Hang on. Oh, nobody's in here. Oh, yeah, they are. If you want to be teachers.
Teachers at this school?
Yeah, teachers. Well, no, some other school. Yes, this school. You can take me literally. I'm not Trump.
Oh my God. It's not wrong. He really is massive.
I don't need anybody to interpret my words in a different fashion.
That lax right there. That's actually true. I'm not gonna lie.
You give me this one. Go ahead. We can have black ones on 2 out of 3.
Okay. See? I'm not even like a job here. I was like, your biogram is.
I see Lytton. Live, love, lab now.
Jack and Jack. What happened when I was going to like?
Oh, very tricksy.
Signs are different. Wow, Mariah, just continuing on your knowledge of science.
Mike, this is KM.
You'll do great.
Really?
I know. Find out. We can.
I like chemistry.
What are you doing? What's going on?
Oh, we're going to first one. I see. So is it that one first? What's the first one? I don't know.
What is this? Like a game?
Oh, outline is final.
Make it make sense to you. Discuss it with your bio buddy. Hannah, you finished? Hannah, what came first? Guys, who's done? Guide me. What came first? Out is what? Out is positive and in is negative. That would be the what? What is that called? Resting potential. Good. What comes next? NA moves in?
No, gates open.
Gates open. All right. Then what happens?
Then the NA moves in.
Then the NA moves in. All right.
And then the outside becomes negative and the inside becomes positive.
All right.
And
then
the gates over.
And then K moves in.
So then the K moves out. And then the resting potential is restored.
Nice.
And then the neuron can fire again. And then you need a long-term fix, so you use the sodium potassium.
Yay. Okay, ask your bio buddy, Shadowlands or Sunshine?
Oh, I can't. Show, I'm here.
Grace, how can you have 4 Chromebooks on your desk?
I like how it took you so long.
Grace, hand a Chromebook to other children. They'll put it away for
I don't
you.
know what happened. Yeah, I think I'm They were like, there's like a huge stack of
Basically, what happened was when I came into class, there was like 2 Chromebooks on her desk, and I was like, you know, it'd be really funny messing with Grace, so I put all the Chromebooks on her desk.
I'm sorry, Grace. Yeah, just start passing them out, and those children will put them away for you. Okay.
No.
Now,
I'll take it. I don't have a
moving on. So going from neuron to neuron is passing on that message, and where 2 neurons meet, that is called a what?
synapse.
Synapse. Good. All right. And so here we can see the axon, right? Electrical signal. Here's the synapse. And the way one neuron talks to another is with the chemical. And they won't release that chemical unless an action potential hits it. And then there are steps right there along the way. So if we look here, here's your neuron. And we know along the length of it, who's coming in? What was that? Sodium, good. So we know sodium is going in, right? Making it positive inside. And then how is that fixed? Potassium goes out. Okay, good. And then, but once you get down to the synapse, it changes. At the synapse, what starts to come in? Something else. Calcium. Boom. Calcium comes in. And when calcium comes in, okay, at the synapse, these synaptic vesicles that are storing a neurotransmitter will migrate, and this is called the presynaptic neuron, and this is the post. Okay, good. And it fuses with the membrane of the presynaptic neuron and does a little exocytosis and releases the neurotransmitter into the space, and the space is called the synaptic cleft. Good. And it will diffuse across, and it will bind to a receptor. This is called a ligand gated. Okay, it's a gate, right? And so what happens is this chemical binds, and when it binds, that's what triggers this protein channel to open. It won't open unless the neurotransmitter binds to it. And when it opens, guess what? It's letting what in? Sodium. That's what. And if enough sodium comes in, it could what the neuron cause? Make it less negative, hit the, and trigger an action potential. Okay? So the neurotransmitter is what opens up the gate for the next neuron. If it is excitatory, it'll open up the gates. But if it is inhibitory, it'll shut the gates hard. Okay? Shut the door. Okay? And so then sodium won't come in. All right. Could blue, in your introductory understanding, explain the little story that's taking place on our screen before us? Go ahead. All right, come back to me. This is a great little diagram to throw. My Flip-A-Pics. This would be a great little diagram to throw in your notes. Either search it, find it. It's from Pearson Education 2006, the synapse. I like it a lot. Because look up here. You can see an action potential number 1 coming in. Then what gate opens up? Calcium. Synaptic vesicles fusing with your presynaptic neuron, releasing neurotransmitter, and then binding. And then it does it stepwise right here on the right. It's a good little review. Did you find it yet? Come on, use your Google. Use your Google powers. All right? So these are ligand-gated, and this picture's in your book, ligand-gated sodium channels as opposed to the voltage-gated channels that the rest are along the length of the neuron. So it's not a voltage that's going to cause it. A chemical actually has to bind before these gates open. All right, and here is a quick little video to show that. Before we start, let's get our bearings. What color is the presynaptic neuron? I agree, yellow. What color is the postsynaptic neuron? Blue. Indigo. This is the whole postsynaptic neuron right here. What part of it would it be? The what part of the neuron? The dendrite. This is a part of a dendrite. What's this space right here called? Synaptic cleft. What do you think the green things are in this picture? Calcium gates. Good. What do you think the purpley things are? Ligand-gated sodium, maybe gates? Possibly, right here, to let sodium in. Okay? So, do yourself a favor. Anticipate what's going to happen. You're more likely to cement it if it actually does happen. You're like, I think this is going to happen next. Okay? Ready?
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated sodium ion channels to open, and sodium diffuses into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
Ask your bio buddies, Shadowlands or Sunshine? Okay, let's put something in our notes. Transmission across a synapse. A synapse is defined as a junction between two neurons. There can be 1,000 to how many? 10,000. Oh my gosh. Okay. The presynaptic neuron is the sending. The postsynaptic neuron is the receiving. The synaptic cleft is the small space between the neurons. Small space between the neurons. Process, calcium gates open, and the axonal bulb of the presynaptic neuron, calcium enters by Facilitated diffusion, exactly. And then you can see calcium fuses. C, neurotransmitters are released from the synaptic vesicles into the cleft and cross it. Released from the synaptic vesicles into the cleft and cross it. And then neurotransmitters bind to the receptors. Either excitation and the opening of sodium gates. Jeremy, bringing it. Okay, or inhibition and a tighter seal on the sodium gates. It is time to search for treasure.
It's tight.
Oh, so I have to start it?
You're literally.
Oh, criminy. I'm going to pause you for a minute. Okay, for those of you that did get the treasure, guide me as I try to get some treasure. What goes first? All right. Then what? Next? I'm looking. Right here. Okay, next? Next? What? Where? What? Synaptic vesicles. Next? Release the neuro. Neuro binds. Treasure. All right. Are you good on this part? Okay, moving on. So the yellow part in here is a cell body, and you can see the dendrites right there, and then a bunch of axonal bulbs. Sometimes they're sending excitatory messages, sometimes they're sending inhibitory messages. There are over 100 different chemicals that act as neurotransmitters. So on your notes, neurotransmitters, 100 plus different known chemical messages that can either excite or inhibit a neighboring neuron. Different known chemical messages that can either excite or inhibit a neighboring neuron. Yes, ma'am?
Where do the neurotransmitters happen? Like, do they just stay
The neurotransmitters are generated and then recaptured and put back and repackaged as synaptic vesicles. And drugs can interfere with their ability to be released, or released too much, or not broken down, or the receptors are blocked. Pretty much all of our drugs either affect the chemical transmission of a gland or a neurotransmitter. And I don't mean just bad drugs. Bad drugs do that too, or anything that's abused. But if you take Advil, right, it's doing the same thing. All right. So different known chemical messages that can either excite or inhibit a neighboring neuron. And then I just want you to know a couple of them. Acetylcholine excites skeletal muscles, but you can see in the notes it also inhibits what kind of muscle? Cardiac. Endorphins are good feelings, runner's high. Dopamines, emotions, learning, and attention. A lot of drugs people take are all about the dopamine. Smoke a cigarette, a little dopamine release. And then if that's how you get your dopamine, because we know as a chemical increases, right, what do we do to keep that chemical in homeostasis? We exert what? A feedback of some sort. Negative feedback. So then our bodies don't make it anymore, so the only way we can get it is by doing our drug. See it? See the addiction? All right. What?
Is that what's called dope?
I don't know that they knew that was the chemical. I don't know. You could do the history of it. Okay? Now, this is in reality what it looks like. Those are a bunch of little axonal bulbs. And like I said, you could have 10,000 of them all over a cell body like that, all releasing a chemical. Sometimes excitatory, sometimes inhibitory, and they need to integrate it to decide. So taking a look at this, tell your bio buddy, would this neuron fire, yes or no, and support your claim? Go. Would this neuron fire? No. Why? Doesn't reach the threshold. Riddle me this. What if there was no inhibitory message? Would it have fired?
Because nothing's stopping nothing.
Look.
Oh, wait, no.
Still wouldn't have fired because even just the straight up excitatory signal did not bring it to what? Threshold. Okay? So when you look at a neuron right here, you can have excitatory messages, see how it's going up, like to, right? Just like the graph we looked at, and inhibitory would make it more negative. And as far as what influences the neuron the most, it's like it's whatever this trigger zone, what it's really called is the axon. Did I talk to you about this? It's like the axonal hillock, and that has a greater vote. Like if I wanted to tell President Trump something, right? So I say it, is it going to have much influence on him? No, but if Steve Bannon whispers it in his ear, could it have more influence? Yes, because he's closer.
He's an advisor.
He's the most powerful man on the planet, but okay. Here you go. I didn't even hear it. I just tuned it out. Like, never mind. It's okay. It's only true that somebody finds out about it. Okay.
It's not conducting a nerve impulse. It's not.
Yeah.
Okay.
I think.
Oh, shoot. Did I forget to restart this? Oh, no, I started it. Okay. He's an advisor.
It's positive, isn't it? Isn't it? During an accident.
All right, guys. That's all the patience I have. You ready? Put in your last moment answers.
I feel like that would be all these things.
All right, how'd you do on this first one? Look at your BioBuddy screen. Do they need a little shame bell in their ears, or did they do an awesome sauce job? All right, guys, let's go through this quickly. Yes. What's the distribution of ions? More sodium outside and fewer potassium inside. Two of you said the opposite. No. Okay. When it's not conducting a nerve impulse, what would you call that? The resting potential. Okay. Repolarization of an axon during an action potential. Who repolarizes? Yeah, potassium. So when let's see. Repeat after me. When sodium goes in, say it. When sodium goes in, that's depolarization. When sodium goes in, that's depolarization. When potassium goes out, that's repolarization. Say it. When potassium goes out, that's repolarization. So depolar, repolar. Right? Got it? I had a really much better one, but I can't remember how I did it. I had it went better. Sodium goes. Do you remember it?
I didn't remember. Well, we never told you. We could make one. Like, have this be like.
I can't remember. Okay, so we'll say it again. We'll say it again. When sodium goes in, that's depolarization. When potassium goes out, that's repolarization. When sodium goes in, that's depolarization. I have no shame. When potassium goes out, that's repolarization. I repolarize you, okay? So when the potassium goes back out, you're going to remember that. Oh my gosh. Let's talk about this. Oh, where are my 9? Raise those hands high. Raise those repolarization. Good job. Okay, synaptic vesicles are at the ends of axons only. Look at you. At the ends of dendrites and axons, what do dendrites do? Receive. You're not going to have any synaptic vesicles there. You are receiving a neurotransmitter. Feel the shame. Feel the burn. Okay? Only at the ends of axons. Check with your BioBuddy and see if they know now, because there's only 9 of you who knew it then. Right? If I'm...
actually really cute. I know. It's like the guys.
Okay, now. Okay, now here along the length of the neuron, where are the synaptic vesicles going to be? Right here. And when they release their chemical, it goes on to the dendrites of the next neuron. So you only have the synaptic vesicles at the ends, because the other one is just receiving it. These are where the ligand-gated channels are, right? On the dendrites. That's where the ligand-gated that receive. Okay, ask your bio buddy, Shadowlands or Sunshine? So transmission of the nerve impulse across the synapse is accomplished by scary McScary. The reception of neurotransmitters at the postsynaptic neuron. Look what some people said. They said A and C are correct. A says the release of sodium at the presynaptic membrane. No. This would be presynaptic, right? This is pre. And this one would be what? Post. Is this guy releasing any sodium? No. No. Okay. So when you go down the length of the neuron, sodium's going in. Nobody's releasing any sodium. In fact, at the synapse, the presynaptic neuron, what's going inside of it? Not even sodium, but calcium. It's the postsynaptic neuron that could have sodium go in, right? But not anything that came from the presynaptic neuron. 4 of you said the release of neurotransmitters at the postsynaptic membrane. Postsynaptic membrane is not releasing it. Who is? The pre. Okay. So, check with your bio buddies, see if they get it now.
It's actually being sent to like.
Did I give you synaptic integration? Peoples? No? Okay. So synaptic integration. The postsynaptic neuron adds up all the excitatory and inhibitory messages of all the synapses and determines whether to send the message as an action potential or not. And determines whether to send the message as an action potential or not. Integration occurs at the axonal hillock, where the axon emerges from the cell body. Closer to the hillock, the greater the vote. All right, so now at this point in your brain, you should understand the evolution of the nervous system and have some ideas about that. Remember when we talked about not having any kind of nervous system at all, just having maybe ions like in a sponge? We talked about nerve nets with Hydra. We talked about 2 nerve cords with Annelida, the earthworm. Talked about a central nervous system and a peripheral nervous system and a mammalian vertebrate system. Then we talked about the structure, the anatomy of a neuron, and all the different parts of it in the myelin sheath. And then we talked about the support cells. Then we talked about how a transmission occurs along a neuron, how it's reset, and the junction between 2 neurons. Right? That's in our head. And as I just reviewed there, I had a review yesterday. What was the review yesterday for? AP exam, right? And we went through the big ideas. And there are how many enduring understandings, my friend? 55. How many enduring understandings did we review yesterday? 1. Okay? So we did 1 of 55. Okay? So we want to work through all of those enduring understandings so you're ready to go for your AP exam. An enduring understanding a day helps you get a 5 on the AP on test day.
There we go.
Okay, so come to the reviews, all right? Now, the sad and wonderful thing about Jeremy is I was thinking that same rhyme that he said out loud, and I would have said it, and it's all of its lameness. Okay, so go to the central nervous system in your notes. Introduction, the functions of your central nervous system. It must receive sensory input, perform integration, and then generate motor output to muscles and glands. Receive sensory input, and then integrate it all, and then generate motor output to muscles and glands. Okay, your central nervous system is well protected by 3 things right here. By bone, Because think about it. Some places in your body, the bone's on the outside, and some places the bone's on the inside. Where are places we have bone on the outside? Around our brain, right? Where else? Right? Our sternum and our ribs. What's inside of our sternum and our ribs? Heart, lungs, pretty important. But look at your arm. The bone's right in the middle, right? And then we're working on that like a lever. So some places on the outside, sometimes our bone's more towards the interior. So we have bone, we have meninges. Meninges are membranes. The pia, the arachnoid, and the dura, okay, are 3 membranes that protect our spinal cord. And we have cerebral spinal fluid that helps cushion it. So I think it's kind of interesting about these membranes because the pia is the closest one right up against the cerebral cortex. Then the arachnoid kind of looks like spider web. And then the dura, very durable on the outside. And look, P-A-D spells pad. Okay? So on your notes, meninges you have, they won't have you memorize those, so you don't need to write it down. I just think it's interesting because it spells pad. Okay. So you can see the brain, the meninges, and then you can see the cerebral spinal fluid bathing it and going all around. Have you heard of spinal meningitis? Okay, that would be when in your cerebral spinal cord, cerebral spinal fluid in your spinal cord has bacteria growing in it. That's really bad. Okay? That's not good. All right. Here we can see our spinal cord, and it looks like a butterfly. It floats like a bee. Okay? This individual is facing this way. Okay? So the butterfly is like this. Do you see this right here is a spinal nerve? Now, all your central nervous system is your brain and spinal cord, and then your peripheral nervous system is everything on the outside. And we have 31 pairs of nerves that go into your spinal cord. They are all mixed nerves. They have sensory fibers and motor fibers. So this is a two-way street right here. But it splits right here, and the sensory neurons are going in the back, synapsing with interneurons, and motor fibers are coming out the front. With me? Did I talk to you about maybe cutting somebody? Okay. All right. So, what? So, any one of you, if I went along your spine right here, if I cut the back, right where the nerves are going into your spinal cord, if I cut along the back right here, I'd be cutting all of her sensory fibers. So she could still move, but she couldn't what? Feel it. If I went around to the front and I cut the front of all of them, what then? She could feel it, but she couldn't move. Okay. All right. So if you look here, okay, here you can see these right here. These are those bony little protuberances that come out right here that are protecting your spinal cord. Here you can see the little butterfly. This person is facing me right now. Here is their spinal cord. Look, pia, arachnoid, dura, the membranes that go around. The whole thing is bathed with fluid. Bones are protecting it. So on your spinal cord, oh, I'm not there yet, but I am there. Let me see the next picture. Okay, here we go. This person is laying face down like this, and we just chopped them, and you're looking down the spinal cord that way. Okay? And, oh my gosh, did I remember to restart this? I did. Okay, sweet. Now, if you look here, where did I tell you about sensory neurons? They go in the front or the back? The back. Here's their little cell body. Comes in the back right here. It can synapse with an interneuron that's going to your brain and spinal cord, and then a motor neuron would come out the front like this, and then the green and the blue would come together as a nerve fiber. Okay? Sensory in the back, motor out the front. Now, do you see how there's gray matter and white matter? Okay? Did I already talk to you about why the white matter is white? There's a covering on it. You know the name of that covering. What was that? Myelin sheath is what makes, okay, where you see it concentrated like this. In the gray matter, there's no myelin. Now I want you to think big. Let's go to the circulatory system. Where does all exchange take place? Capillaries in the middle, right? Because they are what? One cell thick. Remember, you want to make as many synapses as possible You can only synapse and talk to another neuron where there's no myelin sheath, because otherwise the myelin sheath would get in the way. That's why you want to have a lot of gray matter, because that's where all your synapses can be. With me on that? If somebody says, I'm going to scoop something out of your brain, do you want it to be gray matter or white matter? What would you say? White matter. Okay? Scoop out the white, because the gray is where you're making all of your connections. When they talk about the first, if you've ever seen commercials, I don't see them so much anymore. It's called the first four. It talks about the first 4 years of a child's life and how important it is to read to them and talk to them, because that's when all those synapses are going. And if all you do is go, I don't want to talk to my child, I want to do something else, watch TV. Okay, then they're like, okay, they're not making any synapses, not making any connections. That's that whole nature versus nurture. Yeah. What did I do? All right. So, structure, yes. Okay. So structure. Gray matter is cell bodies of motor neurons and interneurons, and it looks like a butterfly. Gray matter is cell bodies of motor neurons and interneurons, and it looks like a butterfly. Whereas white matter is myelinated neurons. Biobody that's blue, why do they call the sensory pathway the ascending pathway and the motor pathway the descending pathway? Discuss.
You're bringing them back to your brain. You're bringing it to your like limbs. You're like moving because he was just for that.
What's the penthouse of the body? The brain. The brain is the penthouse, right? So ascending, you're going taking the sensory information up into the brain, motor down from the brain and out to the body, right? Does that make sense to you?
Yes.
Okay. So what's the job of our spinal cord? Okay, reflex actions, and I'll talk a little more about that in a little bit. It sends sensory info to the brain, motor out, and the tracks cross over. The right side of your brain controls the left side of your body, and vice versa. Now, if you had damage to your spinal cord, depending on where the damage takes place, depends on your functionality. If you damage and dive into a shallow end of the pool, okay, and you break right here and you have a lesion up top, then you're probably going to be a quadriplegic. Okay, a little bit farther down, you're just a paraplegic. Okay, and you have the functionality of your arms, legs. A little farther down, you might need some more help, a little less help. So depending on where the damage is going to determine your functionality. Yes?
So if I get like a stroke on the side to the brain,
Okay, let's say I have a stroke on the right side of your brain, the left side of your face is going to droop.
and then probably left side of my body, I can't.
Yeah, you might have, it depends on where it is in your brain. Your motor neurons are coming out of here, but remember they're all going to different targets from different regions, so it depends where the stroke happens. Yes?
So you generally want injuries like lower on your
Yes. If somebody says, I'm going to take a hammer to your spine, where do you want it? Go low. I want it low. Okay? You know, when they ask you that. All right.
When they're nice enough to ask. So kind.
Now, listen to me. As I've said before, I think I put it somewhere in the notes, like in the summary or something. You don't need to know every single part of the brain and memorize it. So don't stress out about that. But you want to have a working knowledge, because remember, the nervous system is one of your 3 systems. So you better have a good idea about some regions of it and their function. Okay, so I'll go over them in general, hit some highlights. That's why you don't have a lot of things you need to fill in your notes here. Okay, so taking just a general look at your brain, this whole part right here, okay, this is your cerebrum. This is your mammalian brain. This evolved later. And this is all your cognitive functions. This is memory, learning, thought, language, speech, okay? Things you can actually live without, okay? Somebody could take a chunk out of your cerebrum. You might have trouble in school, but you're not going to die, okay? You could live without that, okay? You might not be as coordinated, but you're not going to die. This is this higher learning, okay? Down here, this is your cerebellum, and your cerebellum is, I always think, Sarah the ballerina. Sarah the ballerina is going to have good muscle tone. She's going to be very what? Yeah, coordinated and balanced. And the reason is this part right here, I don't know what he said, but I'm just going to ignore it. Okay. Still ignoring it. Okay, this part right here, your frontal lobe, this frontal lobe is what sends, the frontal lobe is what sends motor information out, and it will do that. But what your frontal lobe would say is, you know, lift your arm, move it to the left. That's what your frontal lobe would do. Your cerebellum would go, you know, and try to make it all graceful and coordinated. Your cerebellum is impacted by alcohol and its functionality. That's why if you get pulled over, they will have you go like this, because you will have a hard time doing that and keeping your balance if your cerebellum. That's why you slur your words. Because if you are okay, like your motor nerves going here to your mouth will say, you know, I like school, okay? But your cerebellum is going to coordinate that so you sound like what we're used to, okay? And fine-tune that. And that's why you slur your words if you've been drinking. That's why you have a hard time walking. That's why they would have you try to walk the line, because you would have a hard time doing that if you weren't balanced. Yes?
So does our cerebellum affect what accent we have?
No, that's going to be your language, and all that's going to be based on memory and what you're exposed to. You know, so what you grew up with. So for instance, if you were from Poland, okay, and you had amnesia or something like that, you're still going to speak with the same accent if you went somewhere else, because that's the only way you know how to pronounce words, right? Okay.
Have you ever like read one of those cases where somebody like goes into a coma, wakes up, and speaks fluent Spanish? Like, what is that? How does that
They
happen?
would have to be exposed to that, because language is memory. So you would have to have been exposed to those words. It's not like you have get superpowers, right? So, but there are all kinds of interesting things. For instance, guys, take a listen. Reception, receiving a stimulus, and perception are two different things, right? I'm receiving a stimulus, but here in my brain is where I'm perceiving what's going on. So you know those images where you go, oh, it looks like an old woman. Oh no, it's two people kissing. Okay, and that's because you're receiving that same message to your eyes, but what you're perceiving it's about is different. It's kind of like when somebody says, why are you yelling at me? And you're like, I wasn't yelling, you know, and you perceive it differently, right? Yeah. All right. Now, come back. Come back to me. Yes.
How does it work if like someone is a savant at something?
You know what? Have you ever known? Because I've known a savant. I knew there was a girl. She was 12. She was blind. She could play the piano beautifully. And get this. And this was at church one time. And I was in charge of turning the pages. and she couldn't see. I know, she was blind. But we were singing. And she was playing the piano, but she could also play the flute. And she would start to play the piano and get so excited, she would grab her flute and play it while playing the piano. And I'm just like, oh, you know, watching her, you know. Incredible. At the same time, her flute, and, you know, she's like got it all propped on, and she's playing. She's 12, okay? Then we were practicing, and the guy who played the bass, she was like always wanted to play the bass. And so she picked up the bass, right? And it's huge on her. She's like, took her about 5 minutes, and she's just messing around with the bass. Then she just starts, and you're like, oh my gosh, okay? In her mind, she was able to perceive the progression and figure that out, and then just apply it. So they are using areas of their brain. They may not know what to do in a social situation, and have trouble perceiving and interpreting, you know, like you watch Sheldon, right? He goes, am I supposed to laugh there? You know, and he just memorizes responses. You're sad, I have to offer tea. You know, it doesn't matter if you like tea or not, you must have warm tea, okay? So they may not be able to interact socially and have that kind of awareness, but their understanding of patterns and themes is so good. I knew another one, a little boy, when he was younger, he couldn't stand jean materials. He couldn't wear jeans, and if I had jeans on, he would say to me, Winnie, why are you wearing jeans? You know, I like them. I think you should wear something else. Okay, but I'm wanting to, but I think you should put on something else. Okay, but I want to wear my jeans. But he had, because his father was a pilot for fun, he memorized every call signal for every airport in the United States. He also liked trains, and you could tell him, you know, what time does the Amtrak go into Portland? 10:25, you know, and he could just tell you just like that. So what areas of their brain they're using, I don't have a clue, you know, but they see and they memorize and make a pattern, not like we're able to do. So I don't think that's well understood, but there are people who, when they're supposed to, they read words, but instead they see colors, you know, and all kinds of ways our brain works. It's incredible. Okay, but I want to come back to this. So here's cerebrum, okay? Here's your cerebellum, about balance. This part right here, well protected, deep here in our brain. So lots of things could happen to us, and this part's still okay. This is your thalamus right here, and then you have your hypothalamus and your pituitary gland. Your hypothalamus is all about this, homeostasis. What are the two systems all involved in maintaining homeostasis? Nervous and endocrine. This is where they overlap. Nervous tissue here, pituitary gland right here is known as the master gland. There's two parts to it, a posterior and anterior pituitary. They secrete different hormones, but they control so many other glands. That's why it's called the master gland. And this is where they overlap. Your thalamus right here, your thalamus does many things, but it kind of connects memory with emotions and feelings. So for instance, if you had like an old boyfriend or girlfriend and they wore a certain perfume or cologne, and you haven't smelled it in a while, and then you're somewhere and you smell it, and you're like, 8th grade. Okay? And it brings you back to that time. Or there's a special cooking spice, and it reminds you of your grandma, you know, and you have memories of your grandma. Whatever that is, okay, those connections are right here in your thalamus. So let's take a look at a couple things here. Okay? So here you can see your frontal lobe of your cerebrum. Remember I said this is where all your thinking is? This is your primary motor area right here. Your parietal lobe back here, somatosensory, your processing area. Occipital lobe is all about visual. And your temporal lobe right here is connecting with your hearing and your perceptions there. So on your notes, go to cerebrum. I gave you all of that. Okay? Your oh, here's some more cerebrum. What do you see there? Gray matter and white matter. Where's the gray matter located? Yeah, on the what? Edges where you have a lot of surface area. See how we can have a brain that fits into a smaller space, right? We have that smaller space because we have all of those what? Folds, which increases surface area so you can have even more connections. In fact, we value the brain so much that when we're born, we can't even hold it up. But we're like, I want it to be big, you know? And it takes a while until you can actually support your own brain, right? Here, I thought this was interesting. I know you can't read all of these, but I want to talk to you about, it says 12 facts you should know about your gray matter. Almost 95% of the oxygen that the brain absorbs goes to its gray matter, and the rest goes to white matter. The amount of gray matter areas in the brain may more strongly contribute to higher IQ than the overall size of the brain. It says, while there are essentially no disparities in general intelligence among men and women, men do have 6.5 times the amount of gray matter than women. Probably just to keep up with us, I'm guessing. And then liar, liar, pantalones en fuego. Huh, liar, liar, liar. I wonder if there's been any liars in the news lately. Oh, no, Sessions would be the news, and he's the Attorney General, but that's okay. He doesn't need to tell the truth. Liar, liar, pathological liars have a deficit of gray matter. Alcohol, chronic alcoholics typically have a reduced amount of gray matter. Anyway, children with autism, remember you were just talking to me? You know, if you're about that, I'm not pointing to you. Children with autism have more gray matter in their brains than healthy children. One study suggests that meditation may increase gray matter. Gray matter consists of neurons, the brain's nerves, we know that, thinking part of the brain. Older smokers lose gray matter and cognitive function at a greater rate than non-smokers. Gray matter is distributed at the surface of the cerebral hemispheres, the cerebellum, and at the depths of the cerebrum, brainstem, and we haven't got to the brainstem yet, but I'm going to show it to you right now. Oh, this is somebody I know. They got in a motorcycle accident. And it's the husband of this woman I knew from this, whatever. This is the picture of his brain. It was smushed due to the injury and inflammation. And then here, can you see? I want to make sure you can see this. It's cool. So I asked if I could have his picture, the pictures from when they were. Look at that. That's not white because it's white matter. That's swelling that was pushing on his brain. And then that's why some people who have had a brain injury that's temporary, they lose maybe their ability to speak and talk, but then they can gain it back once the pressure is relieved. Yes?
Does the brain grow or are we born with the size of the brain?
Born. Okay, now, down into your diencephalon, that is your thalamus right here, your thalamus right here, and then your hypothalamus and your pituitary gland. Hypothalamus is all about homeostasis. I talked about this, about your thalamus. So next to hypothalamus, you need to put the word homeostasis. Homeostasis.
Where's the I don't see it.
Under diencephalon. Okay, your cerebellum, I already talked to you about that. Balance. Okay. And then here, your brainstem. Here you can see your thalamus kind of grabbing on there, weird. Okay, here's your midbrain. That's about relay and reflexes, your pons. And then here, your medulla. I want you to see this because your medulla oblongata, heartbeat, breathing, vasoconstriction, that would be referring to your blood vessels. Reflexes for vomiting, coughing, sneezing, hiccuping, swallowing. So next time somebody sneezes, say, bless you, medulla oblongata. Okay? So that's what's in charge of that. Sorry. All right, this part's pretty interesting, the reticular formation. This tells you to wake up and pay attention. Wake up and pay. And a lot of you could experience this. Let's say Mariah's mother, and she's on the computer, and she goes, Mariah, her brain, if she's like focused on the computer or whatever she's doing, she's like, not important, swipe. And she's like putting it away. And then Mariah, not important. She dismisses it. It's not even in her forefront that she's ignoring it. And then Mariah, swipe, okay? And then her mom goes, Mariah, that, danger, alert, okay? And so she responds, what? And then her mom goes, don't you yell at me. And she goes, you were yelling at me. No, drama, okay? That's your reticular formation, and all the time it's using, it's like when I tell you, okay, talk with your neighbor, and then I'm all, come back to me, and you're still talking, you're going, not important, swipe. It's not until I yell at you or something, and then all of a sudden you're like, and you're paying attention. Think about it when you're home with your family at 9:30 at night, there's sounds around the house, you don't pay any attention to them. You're like, swipe, I'm not paying attention. But you're home alone, 9:30, 10 o'clock at night, nobody else is at the house, and there's a sound, you're like, what's that? And then that's where you would get more nervous, because at that point you're in a different environment, and what you're aware of is very different. Young mothers could be normally hard sleepers, but when they have a baby, their baby will cough, and they're like, and they're aware of their baby. And then if, like, say it's an equal partnership, and so only the woman is getting up in the middle of the night with the baby, and the man isn't, the baby can be crying, and then maybe it's the husband or partner's turn, but wake up, go to the baby. I didn't even hear him, because their brain has thought, somebody's taking care of it. It's not my problem, and they just continue to sleep. People who take sleeping pills take pills to shut down the reticular formation, so that by taking that allows them to continue to sleep. Okay? Then your limbic system. This is higher mental functions with also primitive emotions, memory, and I call this your reptilian brain. Your lower level brain is like concerned with food, sex, paranoia, and when you get down into your limbic brain, you leave your larger mammalian brain when you're drinking and you go down into your limbic brain. Does your lizard brain make good decisions? Not necessarily, okay? So you don't want to live there. All right. Now, to review the structures of the brain, okay? Now, when you click on it, it'll tell you what it is. So try to guess what it is before you click on it. Bless you.
This is a good thing. Spinal, that's the spinal cord.
Did you get it? All right. Speaking of the Fine Arts Assembly. What happens if you don't match it right? You do. I think they play a little song for you if you finish up.
Oh my God, I got one.
All right, my preciouses, come back to me because we need to. I'm not ready. All right. So let's move on, and I can go over that with you in the review if you want to. We have just 3 little boxes left here, and that's about your peripheral nervous system. Remember, a nerve. Look at me. A nerve is a bunch of axons bundled together. That's all a nerve is. Okay, and when you talk about your peripheral nervous system, you have 12 pairs of nerves coming off your cranium. Some are sensory, some are motor, some are mixed. 31 pairs coming off your spinal cord. You can see here an underside. You can see some of the different pairs of nerves. Okay, and here again at your spinal cord, this would be you facing down, slice, right? Sensory neuron coming in the back. And then a motor neuron coming out the front. Okay. And then in between those would be an inner neuron. Let's take a look at this. Okay, and look what this is. It's called the dorsal root ganglion. Ganglion are groups of cell bodies. Sensory neurons, remember how we looked at unipolar? Remember it's not part of the decision making? All their cell bodies hang out right here at the dorsal root ganglion. Here you can see inner neurons, the motor neurons. Here are their cell bodies, and then they're coming out here. Okay? Now, did we look at this picture before? Because it's a great one. Do you remember at the beginning of this topic where we had the central nervous system up at the top, and then we had that long box below? Okay, so this is explaining that as well. So here's your nervous system. Central is brain and spinal cord. Peripheral is outside of it. Peripheral, you have your somatic and your autonomic. Somatic is all about what? Movement, choice, skeletal muscles. Autonomic, we hear the word automatic. And of our autonomic nervous system, depending on what we're doing, if we're going to go play in a soccer game, we need our heart to beat faster. So the sympathetic nerves are going to fire. Okay? If we're chillaxing on the couch, we don't need our heart. We can let it beat slower. So parasympathetic. We want to dilate our bronchi, sympathetic nerves, if we're going to be running around, constrict. If not, same thing with eyes. Dilate the eyes, constrict the eyes, depending on what we need at that time. So on your introduction, go down to somatic system and the reflex arc. Okay? And in this case, here's a TAC. Sensory neuron goes in the back, synapses with an inner neuron, then a motor neuron back out. Why he's jamming the TAC in his hand, I have no idea. Okay? But I've talked to you about this before. If you touch a hot pan, sensory neuron comes in, goes to your spinal cord, simultaneously inner neuron to the brain, motor neuron out, and you pull away before you're even aware that the pan is hot, before you have the perception of hot. You've already pulled away. That's very what of your body? Adaptive. I'm looking for another word. Starts with a P. Yeah, it's protecting. So you don't have to wait for that processing. So somatic system in the reflex arc, the target is skeletal muscles. And then I gave you the reflex pathway. And if you put reflex arc in your search engine, or in Google, it'll give you a good reflex that you can put in there. Okay? And then to the autonomic, if you take a look here, the differences between the parasympathetic and the sympathetic. Parasympathetic, things like slowing the heart, constricting bronchi, getting digestion to take place, bile from your liver because you're digesting, versus here where you're getting ready for stress, accelerating the heartbeat, dilating the bronchi. Look like this. Inhibits flow of saliva. You're getting ready to make a speech and you're like, I need a drink of water. Yeah, you want the water. It's because it's been inhibited. So on the autonomic system, last line, involuntary control of smooth muscles of the internal organs, involuntary control of the smooth muscles of the internal organs, glands, and cardiac muscle. Involuntary control of the smooth muscles of the internal organs, glands, and cardiac muscles. Have a good weekend, and I will see you when? Tuesday, unless I see you for a review before that.